studying the sessions about biotechnology is used in medicine. It's used in gene therapy, and we have seen how the biotechnology is used for the molecular diagnosis. So there are two techniques here. You can see PCR and ELISA. We have studied PCR, polymerase chain reaction. It is used to detect the microorganism or the pathogen in the earlier, in the low concentration in the body. We can detect by the help of amplification of a nucleic acid. This point is very important. It is used to detect, it is used to amplify it is amplification of nucleic acid. So by the help of that we can detect the pathogen. That's by the help of PCR. Now the next one you can see ELISA test. ELISA test, it is one of the important tests for AIDS. What do you know what's the full form of this ELISA? It is enzyme linked immunosorbent assay. Enzyme linked immunosorbent assay. It is used for the detection of the diseases such as AIDS. So how it is possible? It works by the principle of antigen antibody reaction. It works by the principle of antigen antibody reaction. Antigen antibody reaction. So what happens here? Either we can insert the antibody into the patient body. So if antigen is there, then what happens? There will be the reaction with this antigen and antibody, so that we can detect the pathogen in the particular body. Or else what we can understand, if the antigen is there in the body, definitely that antigen, it will produce antibody against the pathogen. So there will be the reaction of this antigen and antibody. By the help of that we can detect the pathogen or the antigen it is present in the body. That's the technique we can understand here, ELISA test. Now one more test is there. Here what happens? Single-stranded DNA or RNA. Single-stranded, a small portion of DNA or RNA tagged with radioactive molecule. It is called a probe. Tagged with radioactive molecule. It is called a probe. This probe is allowed to hybridize in the body. It is allowed to hybridize, and when it is allowed to hybridize, it will make clone in the cell. It will make the clone. It's followed by, it can be detected by the help of autoradiography. So what happens here, where the probe is moving, if any mutated cell is there, if any mutated gene is there, then what happens? It cannot be detected. It cannot be detected in the photographic film. If any mutated gene is there, it cannot be detected in the photographic film. If it is not mutated, then the single-stranded DNA, it will make complementary with the particular strand. So by the help of this technique, we can identify by the help of autoradiography whether the gene is mutated or not mutated. So this is the earlier detection, some of the techniques which are used for the earlier detection of the disease. Once we find detected the particular pathogen or the disease, immediately we can go for the treatment, so the cure also will be very easy. So this is about the molecular diagnosis. Now next we can go to study, we can study about another topic, that is called transgenic animals. It is called transgenic animals. So by the help of the word we can understand transgenic. Already we have discussed genetically engineered plants, genetically engineered microorganisms, genetically engineered animals. Here you can see transgenic animals. What you can understand about the transgenic animals? The animals, their DNA or the genetic material is manipulated, or it is altered, or it is changed. Such animals you can say transgenic animals. We have studied in the last chapter biotechnology and its process. There are two principles. Are you remembering? There are two principles, that is genetic engineering and bioprocess engineering. In the genetic engineering we have studied the particular gene we have to extract and we have to insert into the another animal, so that that animal can express, it will have the phenotypic expression. So what here you can understand, changing the genetic material, altering the genetic material, manipulated manipulate the genetic material, that only you can say transgenic animals. So there are animals, some animals we can use for the transgenic animals, transgenic rat, mice and pig and mice. There are many animals we can use for this purpose. Mostly mice using in the field. So how it is used, why it is used, here we can see some of the purpose of these transgenic animals. There are some purpose of the transgenic animals. First let us see what is the first purpose of this transgenic animal. So first one is normal physiology. Normal physiology of an organism. Normal physiology of an organism. What happens here, when we insert the gene into the particular organism, what will happen, because of this gene we can understand some of the foreign particle, so it may affect the physiology of an organism. So how it is affecting, whether it is affecting or not, if any affection is there or not, how it is affecting in different physiology like digestion, respiration, nutrition, whether it is affecting or not, that we have to study. So that only we can understand normal physiology of an organism. We have to understand whether it is affecting of the body, so that we have to understand, that only we can say normal physiology of an organism. And the second purpose we can understand, study of disease. Study of disease. So the transgenic animals we can use to study certain diseases. What you can understand means there are some genes are responsible for some of the diseases. So if we find a particular gene is responsible for the particular disease, then it is very easy for the treatment of the disease. So we can understand, we can go for the treatment of the disease and prevention of the disease. And not only that, there are some transgenic models are there. Some of the diseases like cancer, cystic fibrosis, some of the disease, that models, human models, they have made with the transgenic animals, so that immediately they can study which gene is responsible for such diseases and what is the treatment, what is the prevention of the disease, we can study by the help of transgenic animals. So next, one of the purpose, another purpose we can understand, biological products. Biological products. So what you can understand here, some of the biological products, some of the treatment, medicine for the treatment of the disease, that biological product for the treatment of the disease is very expensive. It is costly. So what you can understand here, so if we identify particular gene is responsible for the product, that gene is inserted into the particular organism, then immediately the product can be made. So it can be easy. So that only we can understand about the biological product. So this biological product and two more purpose is there again that we can study in the next session, in the next video. Please watch the next video also. Thank you, students.